Welcome back, students, in our this assignment, and this is your assignment number 2 of chapter Plant Life Photosynthesis. Now in this assignment I am going to teach you about the stomata and the mechanism of opening and closing of stomata. Now first we start with the stomata. Stomata are the microscopic openings on the surfaces of plant leaves that allow for the easy passage of water vapors, carbon dioxide, and oxygen. Now, children, the stomatas are the tiny microscopic openings, or we can say the tiny pores that are present on the surface of the plant leaves, and it allows the water vapors to pass through them, and it also allows the carbon dioxide and the oxygen to pass through those tiny pores. Now, they are crucial to the function of leaves as photosynthesis require plenty of carbon dioxide as well as release of waste oxygen for plants and excess of water vapor. Now, stomata means the mouth in the Greek language. The main site of stomata is on leaf surfaces, but they are found on all above parts of the plant. So the main site where the stomata are present, it is present on the leaf surfaces, but they are also found on all above parts of the plant. Now the number of stomata that are present in a plant depends on the environmental conditions such as higher light levels and moisture or lower carbon dioxide levels. So the number of the stomata, it depends on the condition, on the environmental conditions. Their number increases in better environmental conditions to maximize the rate of photosynthesis. In warmer environments and particularly in dry air, plants lose a great amount of water through their stomata, and in such conditions stomata closes and rate of photosynthesis decreases. So this is done by plants to save their water reserves. So this exchange of gases and this exchange of coming out of water for extra water from the pores of the stomata is because of to save the water. The excess water comes out from the pores and it saves the water for the plant survival. Now we see the mechanism of opening and closing of stomata. Now the opening and closing of stomata is regulated by turgidity of guard cells. Now turgidity means, children, the swollen due to the pressure from inside. They swell because of the pressure from the inside. The stomata are open with the guard cells when the guard cells are turgid. So when the guard cells they are turgid, the stomata open. On losing the turgidity in guard cells, the stomata are closed. So it depends upon the turgidity condition that when the stomata open and when the stomata close. Now here you can see the diagram of opening and closing of stomata. In the first diagram A shows the guard cells, the turgid guard cells, and the stomata opened. So when the pressure applies from the inside, the stomata opens, and when the pressure when the turgidity is loses in the guard cells, the stomata are closed. You can see the B part, the flaccid guard cells and stomata closed. Flaccid means that the dropping or lacking of water, and because of this dropping and lacking of water, it reduces its size, it loses its size, and because of this they shows like that the stomata is closed. And you have to draw this diagram in your biology copy as well. Now you can see here the nucleus is here, the thickened wall is there, and this part of the stomata is known as the guard cells. You can see this black part, this is known as the guard cells. Now we can see here the structure of stomata. Now each stoma has two bean-shaped guard cells. You can easily see here the structure of stomata, and you can see here the guard cells. Then the inner thick cell wall is there. Then epidermal cell is here, and nucleus. Here you can see the nucleus is present over here. Stomatal pore called stoma. This pore is known as stomatal pore or stoma, and these green dots, they are known as chloroplast. So each stoma has two bean-shaped guard cells. They have inner thick walls as compared to the outer walls. These inner thick walls are thicker as compared to the outer walls. When the guard cells are full of water or turgid condition, they swell and bulge outwards and the stoma opens. Now what happens is that when the guard cells, they are full of water, so because of that fullness they swell up and they bulge outwards, and that in that condition the stomata opens. Now see the next picture, stoma closed. In this you can see the stoma is closed because of the loss of water. When the guard cells lose water or they become in the flaccid condition, they lose their turgidity and the stoma closes. Children, this diagram also you have to draw in your copy, the structure of stomata. Now we see the functions of stomata. First is stomata helps in the exchange of gases between the leaves and the atmosphere. As we all know that the exchange of gases like carbon dioxide and oxygen, it takes place here in the stomata only. So it is responsible for the exchange of gases between the leaves and the atmosphere. Second point is it helps in the process of transpiration. So this process is transpiration is a process in which the excess water comes out from the plant through the pores, through the tiny pores that are known as stomata. So this is all about the function. Now there is an activity for you. You have to see this activity, you have to understand it, and you have to write it in your copies as well. Now this is activity 1. This is activity 1 to demonstrate that carbon dioxide is necessary for photosynthesis. For this, the requirements are 2 similar potted plants, 2 bell jars, watch glass, sodium hydroxide, sodium bicarbonate, corks for closing it, delivery tube, etc. Now the procedure is take 2 similar potted plants which are nearly of the same size. In this activity you have to take 2 potted plants which are of similar size and which are similar as well, which are same. Second is keep them in a dark room for 3 days to de-starch them. De-starch means the removal of starch from the leaves. Now cover both the plants with bell jars and label them as A and B. So you have to label those potted plants so that you can easily, you can clearly note down the observations. Now inside the setup A, keep sodium bicarbonate and it produces carbon dioxide. Then inside setup B, keep soda lime. It absorbs carbon dioxide. So in one case it produces carbon dioxide and in second case it absorbs carbon dioxide. Now what you have to do, use Vaseline to seal the bottom of the jars to the glass plates so that the setups becomes airtight. Then keep both the setups in the sunlight for at least 6 hours. Now perform the starch test on both of the plants. Now what do you observe is observations. The leaf of plant B, which contains the soda lime, remains colorless, showing the absence of starch. So the one with the setup of B, that contains the sodium soda lime, and it shows colorless. The leaves are colorless. Then the leaf of plant A, which does not have soda lime, it turns blue-black, showing the presence of starch. So the setup A, which contains the sodium bicarbonate, you can see that the leaf which contains which have this which have the sodium bicarbonate, that plant leaves turn blue-black in color, and this blue-black color appearance shows the presence of the starch. Now the result, since the carbon dioxide was absorbed by soda lime in the case of plant B, therefore photosynthesis does not occur and there is no positive starch test. But in second statement, the normal photosynthesis process takes place in plant A in the presence of carbon dioxide. This means that carbon dioxide is necessary for photosynthesis. In setup A, the leaves color turn blue-black in color, and because of the presence of a starch, so we can say that the carbon dioxide is necessary for photosynthesis process. Now that's all about this session. Soon I upload the next video for you all. Thank you.